Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on basic biological tools. In our last lesson, we learned about the scientific method. We learned that it is the process in which scientists approach their work. We looked at the steps involved in the method, and we also discussed the work of biologist Francesco Redi. He conducted the first scientific experiment to disprove the idea of spontaneous generation. Today, we begin a series of lessons on basic biological tools. Over the next few lessons, we will identify biological tools, classify the use of each tool, and determine the specific function of each. Are you ready to get started, students? The types of tools and equipment that biologists use to conduct research and experiments are vast. Not only are there many different kinds of tools for various uses, but these tools come in a variety of sizes and shapes as well. Students, make a list of as many biological tools as you can think of. Try to think of different jobs biologists carry out and what tools they would need for each job.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I bet you were able to name a few tools. You may have listed test tubes, balances, cylinders, and magnifying glass, just to name a couple. There are many different items that are used by biologists. We could spend several hours if we look at every tool or piece of equipment that they use. But instead, we will only learn about the field and laboratory equipment that biologists everywhere would recognize and use most frequently. In the laboratory, biologists use the following basic tools: microscopes, dissecting equipment, petri dishes, pipettes, syringes, centrifuges, measuring cylinders, and balances. Cylinders, pipettes, and syringes are tools utilized for accuracy. They are used for measuring volumes, usually of liquids, but sometimes of gases. Balances are used for measuring mass. They can be found in a range of sizes and properties. Some balances can measure the size of extremely heavy objects, while others can measure the mass of an object to the nearest 1/10,000th of a gram. Biologists often dissect specimens to observe their insides. Dissection means to cut apart or separate tissue for anatomical study. The whole organism can be dissected, or just parts of the organism, such as the heart or kidneys. Dissecting can give information about what an organism eats, how its internal structures are built, and how the structures are connected. A standard dissecting kit contains a magnifying glass, scalpels, scissors, tweezers or forceps, and mounted needles. Scalpels are used to cut away the skin of a specimen. The scissors are used for any additional tissue or material that needs to be separated. The tweezers and needles are used to manipulate the specimen. The magnifying glass is used for closer observation. Students, before we continue on with the lesson, let us do an activity to confirm our knowledge of the material. What are cylinders used to measure? What are balances used to measure?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I trust that was a simple activity for you. Cylinders measure volume and balances measure mass. Did you write down the same answers? If so, great work. Let us carry on with the lesson. Petri dishes are round dishes made from glass or plastic. They have many different uses, but their main function is to contain culturing organisms. Petri dishes are often used in combination with agar. Agar is a jelly-like substance obtained from seaweed. It is used for culturing microorganisms. There are many varieties of agar, and each contains a different balance of nutrients. Therefore, each type of agar encourages the growth of different microorganisms. There are two main types of microscopes used in biological work: optical microscopes and electron microscopes. Optical microscopes use beams of light to produce magnified images. Electron microscopes use beams of electrons to produce magnified images. Centrifuges are used to separate solids from liquids when simple filtration will not be adequate. The mixture is placed in a centrifuge tub and placed on the centrifuge. The machine then spins the tubes and gravitational forces push the solids to the bottom of the tube. An example of how centrifuges are used is to separate various plant and animal cells. A quadrat is a tool used to estimate how many different organisms there are in an area. There are many different types of quadrats, but the simplest type is a square metal frame. The quadrats are placed randomly in an area, and then the number of different organisms are counted and recorded. This not only measures the different organisms, but also how species change across an area. A few other common tools that biologists use are data loggers. These are used to record information. pH kits are used to measure the pH balance of soil or water. A flow meter is a tool used to measure the rate of flow of water. A theodolite is a tool used to measure the height of trees or of slopes in an area. The most recent addition to the list of common tools used by biologists is a Global Positioning System, or a GPS. This equipment makes it possible for biologists to record positions quickly and make accurate maps of area of study. For our final activity, students, list the tools that we talked about today that are used to aid observation. You may begin now.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, everyone. There are a few tools that we learned about today that can make observation easier. You may have listed a microscope or a magnifying glass, or perhaps you said Petri dishes because they allow biologists to view bacteria or other microorganisms. You may have also said any item from the dissecting kit. All of the items function to remove the overlaying structures of a specimen to view specific structures closer. If you listed any other tools that I did not mention, please review your answers with teacher at the end of this lesson. This brings us to the end of our lesson on biological tools. We have covered quite a few tools and pieces of equipment today, ranging from items that measure to items that observe. I hope you enjoyed this lesson. Until next time, thank you, teacher. Thank you, students.